enough, and I got a chance to do my PhD in the UK. And once I was there, I got a chance to visit a local school. And one day, the teacher was trying to teach the students, or young boys and girls, to recognize even animals. And the teacher just picked up a picture of a deer. You know the animal deer? That's an animal? Okay, reindeer, the deer. And ask the boy. And do you know the name of the animal? The boy looked at the picture with a confused expression on the face and said, "No, I don't know." And the teacher was not quite easy to give up, and she tried her best to make the boy to understand the name of the animal. And he asked, "Well, how's your mom call your dad at home?" And then the boy brightened up and said, "Yeah, I know that." And then his face turned to be even more confused expression and said, "Madam, are you sure it's a pig?" Okay, that's a story just to make you laugh and to make you feel not so bad and not so tired. And then we can continue with the biotechnological reading. And this is the work also being done here in the University of Costa Rica, where I read from the poster on the wall. And even though I cannot read anything in Spanish, but still I noticed the pictures and I realized students here are doing this kind of work here. And protoplast fusing. The protoplast means the cells, the plant cells with no cell walls. And without the constraints of the walls, the cells appear to be the round like balls. So that's called the protoplast. And to fuse the protoplast together from two different species and to push them to link together and then to mix the material inside the nucleus and to put them together, which is quite similar to the process we call the hybrid. And since the cells are taken from the tissue of the such as the leaves, etc., and this process is called the somatic fusing or somatic hybrid. That means the body cell rather than the sex cells. Okay, understand that? That is the process of the fusing. You push two different cells together, and then the mixture of the inner part of all the materials, and then actually they will regenerate into a new plant. And that's the example just to show. For example, you have a variety with the red flower, and you pick up a cell and take off the cell walls and produce the protoplast. And then the next one with the purple or the blue flowers, and then you do the same thing and mix them together and ask or make the cells to fuse together and to produce a hybrid, a somatic hybrid. And that's the process, or not process, the procedure, you can say, to prepare for the protoplast, and then to fuse the cells, and then regenerate the cell wall, and induce the colors, and then to induce the hybrid. And nowadays in China, our breeders or scientists have tried everything, well, quite a lot of things, not everything, quite a lot of to cross-pollinate, not cross-pollinate, just to do the somatic hybrid between species, such as between carrot and celery. We know in nature that sort of cross-pollination can never happen because the two species are totally different. And also the hybrid between kale and cabbage, and also watermelon and muskmelon, and also radish and cauliflower. And some results are very interesting. So, such as the cabbage kale, a different species or a new species produced by the somatic fusion. And but I don't want to put something in your mind. We can realize something like that. The hybrid, now the somatic hybrid between the carrot and cauliflower will give you something like that. Now, I don't think that will happen. But from the fusing, possibly it is a method or it is a way for us to introduce some certain genes from one cell into another. Most mostly the most used genes for scientists to look at is some sort of resistance genes, such as the blackboard resistance in cauliflower, and can be transformed using this method, well, from one species into another. And also, we can use that method to transfer the resistant gene from one species into another, such as the resistance to bacterial wilt in potato. And Chinese scientists have now introduced quite a lot of resistant genes from, for example, from the wild eggplant into the cultivated eggplant, through that method and with the enhanced resistance. So that's the achievement Chinese scientists have already got. And also, people from Hubei province have tried to transfer the CMS gene from one species, from one cultivar into another cultivar of oilseed rape. But it's quite weird because this sort of hybrid can be realized from the cross-pollination, because it's from a species within a species, just between two different varieties. So, but this is just an example of how we can use the somatic hybridization to do the breeding work. And we can also start the tissue culture or cell culture from the microspores. Not knowing whether you've tried something like this, but it is a very useful tool for us, for the breeders, because for the microspores, which means, for example, with a diploid, which means in the cell there will be two sets of chromosomes, but in the microspores, only one set of chromosomes existed. Is that right? And if you can just regeminate a plant from the microspore, which means that plant will be with only one set of chromosomes. And once you double the chromosome number, you will have a very pure diploid lines easily. For some species such as the canola or that sort of thing, because that's normally the cross open pollinated, and it will take the breeders many years to get a pure line. But in this case, you can easily get a pure line within only one generation of the planting. And for some, yeah, that's it. And so far, starting from the 1970s, and now China has developed hybrids from over 40 vegetable species, such as broccoli, lettuce, radish, carrots, peppers, blah blah blah. And also the introduction of peppers with a cabbage hybrid was first succeeded in China. So that's the thing our Chinese scientists have achieved and contributed to the vegetable breeding. And that's the procedure. We can say, firstly, we will collect the immature, the microspores, and then culture it, and then to induce the embryo. Embryos can then be induced into a whole plant. And, but we may know all the plants are haploid, which means have only one set of chromosomes in their cells. And if we want that sort of plants to bear any seeds, we need a step which is called the chromosome doubling. To double the chromosome number, to make it from haploid into diploid, and then to produce the seeds. And that's a chemical from a nature herb, which is called the colchicine, from a species of white flower. And that flower looks very beautiful and is also edible, can be eaten. But we must be very careful. And the fresh ones are toxic. If you dry the flowers, you can eat it. And to use that chemical, that's the function there, and how we can double the chromosome numbers. That's the information you can get from any textbooks, and it's called the spindle poison, which means from that part, the chromosomes will move to two different poles of within the cell. But if the chemical can cut that way, and the chromosomes will not move to the opposite sides of the cells, and then the cell won't divide into two cells, and then in that single cell, the chromosome numbers is doubled. And Chinese scientists have already innovated the paper haploid breeding method, and to reduce the haploid, that's the work done by people from Beijing. And since 1982, and Beijing Laboratory of Plant Tissue Culture and started its studies on inducing the paper haploids. And with this technique, series of haven paper varieties have already been bred and released. These are the two examples of the newly bred varieties following that method. And the Institute of Vegetable and Flowers always won the national prize in 2006 for their work on this part. And that kind of work has been used to induce the variations. Because the media, whatever media you use, that's not the optimal situation for cells or for organs or tissues to grow, because that's the man-made media. So some sort of things or chemicals will be not, that sort of inducing, which can induce some sort of mutants. And with that sort of works, if the mutant gene is in the haploid, and after the chromosome doubling, you have a pure mutant mutant. And with that work, that's the work has been done by Zhejiang, people from Zhejiang province. And also in cabbage, or scientists from Hunan, they have produced different varieties following that method. And also their achievements won the national prize in 2006. And with that method, there's another interesting thing we can imagine. We can produce the seedless watermelons. And I'm not sure is it very common here in this country, but it's very common in China, because Chinese people are not very, happy with the seeds inside the fruits. We just want to eat the melons You do not have to worry anything about the seeds inside. So if we produce a watermelon variety which can give you only the fruits without seeds, you can have a better market, and people like that sort of fruits. And that technique firstly introduced from Japan in 1951. And the procedure of producing the seedless watermelons is just like this. Firstly, we treat female parent, which means that the haploid, and to treat the flowers with the courtesy and produce the diploid. That's a tetraploid with 4 sets of chromosomes, and then crossed with the male parent with the parent with only 2 sets of chromosomes, we produce the triploid seeds with 3 sets of chromosomes, and then the company will sell the seeds to the farmers. The seeds will germinate into a plant, but unlikely the plant will bear any fruits, because the 3 sets of chromosomes will do nothing for the cell to divide, and in that case, no fruits will be there. And in that case, farmers still need to plant several plants, which is called the pollinizer, to produce the pollens, and the pollens are used just to induce the formation of the fruits. And in that case, after the induction, the fruits will be there, but no seeds will be inside the fruits. So that's the procedure of produce the seedless watermelons. If you are interested in this, you can just copy this picture. Okay. And now there are several very famous examples of varieties, of seedless varieties from the Jiangwei company, such as the idea number 2 and idea number 1. Looking back, just as I said in the early morning, breeding is looking for the difference. And in nature, there are differences induced by many different factors. And if we scientists or breeders are now willing to improve or enhance the induction of mutants, we can use a method which is called Mutagenesis, which means we can use different things to induce the mutants and then to select the ones we are interested in. And the factors to induce the mutagenesis are called the mutagens. And there are chemical mutagens, or physical ones, or even biological ones. And for physical mutagens, if you have the interest to do such work, possibly you can try to find out whether you have access to X-rays or gamma rays or the other chemicals which can cause the DNA damage. And also the UV light radiations, and also some kind of radioactive decay. So if you do want to do such work, you need to look around you and try to find out what kind of mutagens you have. And also for chemicals, there are some sort of DNA reactive chemicals, such as ROS, the reactive oxygen species, or EMS. Is a chemical very widely used in the molecular laboratory, EMS. And also there are quite a lot of other things, such as the Bath analogs, and also some of the heavy metals can have the potential in inducing the mutants. And for the biological ones, such as the transposons, and virus, and even some bacteria will do that work. And This gives you a very brief review of the mutagenesis, such as for the physical mutagenesis, it started from early 1920s, and first it succeeded in inducing the mutants in fruit fly. And that discovery, when the people, the Nobel Prize 20 years later. And also lately, that technique was adopted in inducing the mutants in maize and barley and the other crops. So this is the information, basic information you can just follow. And later on, such as in tobacco, barley again, and all this information. And in China, not in China, and since 1960s, with the development of nuclear technology and physical mutagenesis in crop breeding was highly promoted. And since the year 2000, and in the new era, in the new century, the space technology was developed and was applied to crop breeding, which come out with a new word which is called the space mutagenesis breeding. That's the thing might not be available for all of us, but because that depends on the satellites you launch into outer space. If you don't have the satellite, uh, it's not uh, it's not possible for you to do that. But in China, that's quite convenient because we launched quite a lot of satellites into the outer space. For the chemical ones, also started from 1940, and we use the most of the gas. That's a very toxic gas, and widely used during the wars, and it won't kill people, but to make people feel ungood, not good. And also the other chemicals, and calculated at 2012, and in the world there are more than 200 countries developed more than 3,000 mutant crop varieties. Among them are 376 varieties induced by chemicals. And also in vegetables, they are covering 49 species with 423 mutant varieties bred by this method, and 20% of them are induced by chemicals. So from here, from the data, you can say that mutagenesis is a widely used method in vegetable breeding to induce any difference of mutants. So if you are a breeder here, possibly you can think about it. And in China, this method started at the late 1950s. And so far, there are more than 50 institutes adopt such method in their breeding. And so far, we have accomplished also with the other method, and the Chinese scientists now have bred 625 mutant varieties covering 42 crops. And most famous examples are in rice, cotton, soybean, wheat, and groundnut. And the varieties bred following this method have been cultivated over 9 million hectares in total. And a large number of mutants or germplasms have been developed and stored in different institutes. And here are some examples of how many varieties we got following this method. For example, for vegetables, during that past 40, less than 14 years, we have already produced 16 varieties bred by this method, the mutagenesis method. And most widely used is on cereals such as rice and wheat. And now there are several institutes have developed their own treatment chambers. For example, for Zhejiang Academy of Agricultural Sciences and Heilongjiang Academy of Agricultural Sciences, they have already established a gamma ray treatment chamber. So that sort of equipment make the breeders easier to use or to adopt that kind of method into their breeding program. And just a quick example of the effect of gamma ray treatment on sweet potato. You can see the leaf size and shape, very wide variations. And also for the fruit, you can see different size. So you can just pick up the one you are interested in and then carry on the normal procedure of breeding. Luckily, several years later, you will have a new variety. And for example, a soybean variety, which is called the Cushionoid, was released in 2004 by this method, and with highly increased virus resistance. And that kind of work was done by the Chinese Academy of Science, which is in Beijing. And about space breeding, and starting from the 1987, China initiated a project sending crop seeds into the outer space. The first purpose was not to produce any mutant varieties or for breeding. And the initial purpose was to evaluate the influences or impact of outer space on the genetics of plants. Just trying to find out what will happen, because once we send people outside, into outer space, we need to know that kind of conditions might have some sort of influences on human body. So we tried first with the some of the seeds, and unexpectedly, scientists found that some interesting mutants appeared from the recovered seeds. And then they realized it might be a very promising and useful method in breeding. And then in 2003, the government started a project on space breeding. And so far, we have already launched over 70 crops into the outer space and released more than 30 varieties, including rice, wheat, cotton, tomato, paper, and sesame. In total, we have launched more than 100 satellites into the outer space. And many of the satellites carry with them some of the seeds. Here are several examples. So left, that's a new variety bred, or new watermelon variety bred following that method, which is function number 1. Hung means that sort of, outer space, that sort of thing. That's the Chinese word. And also the right side, a new variety bred of the paper variety, and with increased yield. Which is that the fruit weight increased from 90 grams to 170, nearly doubled. And also with the vitamin C content increased by 20%. And the yield increase, average yield increase around 30%. And from that part of work, we normally, if we have already sent the seed to the outside world, to the outer space, our focus on selecting is normally the bigger ones. So it is a very common phenomenon in China for people to understand about the space breeding. They would like to think the bigger ones are induced by the outer space conditions. So maybe that's not true, but that's there can be some sort of misunderstanding by the normal people. Because, you know, in the outer space, you know, the gravity and the radiation, you know, everything changed. And even for the astronauts, when they come back from the outer space, their body can be a little bit taller. So, you know, well, that's the way we consider the space a brief. We are looking for the bigger ones. And that's the reason why the varieties bred following that method can have higher yield. Okay, the last part, we can move on into the molecular breeding. And the molecular techniques adopted in plant breeding included the marker-assisted selection and also the transgenic breeding. And for the marker-assisted selection, MAS, if you are well careful enough, you may remember William, I think his name is William yesterday, had already covered some of the work with MAS. And for my part, it's quite easy to understand. For example, if we are trying to do some breeding work to produce a new variety of apple, to use the MAS method. For example, say you have 100 seeds with you in a pocket, and there might be a different shape or different color of apples. But in your project, you are only interested in the ones with the purple color. So in that case, for the traditional way, you must sow or plant the whole seeds into the field, and years later, you will harvest the fruits. And then you try to cut the fruits into halves and try to find out which plant is the one you are interested in. So that's a traditional way of selection. And you know, you need many years and a big land to plant all the seeds, and most likely, most of your energy is wasted because you're just looking for something with interest. So in this case, the marker-assisted selection can help you. And you can germinate all the seeds and pick up some tissue, say the leaves, and extract the DNA and run the PCR. And from that, with the linked markers, you know, that sort of markers should have already been established. And you choose only the ones with the markers, and you say, yes, that will be the thing I'm looking for. And then you can just need to grow only 2 plants or trees rather than all the plants. So that will save you a lot of money and a lot of time, and to make the selection very efficient. And also yesterday, there's a question asked to William about the QTL markers, and which means if we can use the QTL markers in our breeding program, I would like to say yes. And if you have enough QTL markers and you choose the markers with the targeted traits, and then you select from the later generation only with the markers, and then you will improve your efficiency of selection. Luckily, scientists now have already built up the linkage map for many different species of crops. That sort of linkage map will help the breeders to choose the right markers. Without these sort of markers, you can do nothing with MAS, because the markers are the fundamental step for us to start with. And also, the SSR linkage map of common B. You can see quite a lot of different markers there, and if you say there's a 1G there with the interested traits you are interested in, and then you can pick up the markers around the loci, and then in your breeding procedure, you just select the offsprings with the linked markers. So that's an easy explanation of MAS. And that of linkage map are now also being done in China, and the Chinese scientists now have already finished the genomic sequencing in cucumber, cabbage, and watermelon, pepper, etc. And this is an example of what we are doing by the MS method in the breeding of a pepper variety, which we call the Zhongshu number 12. And from the normal traditional cross-pollination method, we the upper part, and then we have got the different lines to make the process between the two lines, and then from the offsprings, we need to choose. Well, because at that stage, the breeders must have a lot of different seeds, and they do not want to sow all the seeds, and then they use the method called MAS and select the seeds only with the markers they are interested in, such as the virus resistance or nematode resistance or the disease resistance. All with these markers, they kept the seeds. And then they developed a new variety with increased resistance to all these pests. And also, there's another example achieved by Chinese breeders or scientists. They introduced, they breed virus-resistant tomato variety following the method of MAS. And cucumber variety, and also the same to select the ones with the markers linked to the resistant genes of certain diseases, such as the powder mildew. And that's the MAS part of the work, because William had already discussed that part to you yesterday, so we just need to go very quick. And now we are looking for something which might be interesting to you, which is called the transgenic breeding, and which is also called the breeding by design. We are playing God, so in that case. Transgenic technique serves to introduce gene sequences for expression of a desired trait. And the production of transgenic plants is reported in many crops, but commercialization is limited to a very, very limited crops, such as cotton, especially the BT cotton, and the corn, also BT corn, and soybean, with the resistance, tolerance to some sort of herbicides, and also canola. So that's the fact of background, and with the rapid development of modern techniques or technology, scientists can now manipulate the gene, which means they can operate and they can design the gene according to their needs. And even though GMO products now in China are not very warmly welcomed, but still Chinese scientists have already succeeded in getting transgenic plants in more than 35 families of species, or covering 120 species of plants. Now, among them are some very important vegetables, such as tomato, potato, carrot, and other varieties, and the other vegetables. And the genes transferred are mainly covering these sort of things, such as disease resistance, pest resistance, herbicide resistance, and also for some fruits or vegetables to transfer the genes which linked to the elongated storage and quality, or even the male stereo genes. And if you have the experience or understanding of GMOs, it's quite clear for you. It must be very clear for you how we can produce the GMOs, and that's one of the key steps is called the gene transformation, which means you must transform a gene from one species into your crop. And normally, for us to do the GMOs, the genes, the alien genes, normally come from mostly from bacteria or even some fungus. And the well-known example is the BT gene, and you can just oh no, that's not from this one. This is just a transformation procedure. And we got a bacteria, which is called the agrobacterium, and then there's a T plasmid inside the bacteria, and the part of the genome in the plasmid can be cut off, and then you just insert the new gene or the target gene inside that part of plasmid, and then after the transformation, the gene will be linked into the plant cell, and then to produce the transgenic plant. So that's the overall procedure of the transformation, and mostly used for the first successfully used gene, including the pest-resistant gene. And the most famous example is the BT gene. And the BT genes come from the bacteria which scientists collected or isolated from the soil. And that kind of gene can produce a BT protein. And that protein, once eaten by some certain kind of insect species, and that protein will kill the insects. So, such as the insects from the cotton or canola or even rice. And so, the BT transgenic crop will have the natural resistance to such kind of insects. And this is the BT gene, how you can transfer the BT gene into maize. And the first very widely commercialized crop of GMO crops, that's the maize, succeeded from America. And the BT gene from a soil-borne bacteria put the BT gene into the plant. And once the insect eats the plant, the BT protein will kill the insect. And that's the example. And with the BT gene, you can say the ears won't be affected by the insect. Because once the insect eats part of the ear, they will die. But for the untreated control, you can say many of the seeds have already been eaten by the insects. And also the BT transformed canola or oilseed with the gene or without the gene. The differences are very, very obvious. And for BT potato, you can say affected. That's the control affected by the insects, and the BT ones very clear. And for cotton, BT cotton with the BT gene and without the BT gene. So you can say differences are very, very clear or significant. And in the year 1991, the Chinese scientists got a BT cotton plant all by ourselves. And then several years later, the BT transformed in Chinese cotton variety have already been used. And also in 1998, and we have BT cotton released and commercialized in China. And till now, till 2007, and over 10 GMO cotton varieties have already been released, covering over 80% of all cottons produced in China. So the BT cotton is now no problem in China. And also with the modified BT genes, and the Chinese scientists have already produced the BT modified varieties in cabbage, in cauliflower, and also in other vegetables. And the second widely used genes are called the herbicide-resistant gene, and which gives the plant some sort of resistance to a herbicide, which is called the glyphosate, which is with a commercial name of the Roundup. And to transform the bar gene into the crop, and the transformed crop will have the resistance to that sort of herbicide. And then after spray the herbicide, all the other herbs or even the weeds will be killed, but the transformed crop may survive. So that's the basic idea about why we use the BT. No, the bar transformed gene. So that picture shows with the gene you spray the herbicide, and all the other weeds and the other plants have already been killed, but only the GMO rice can survive. And also for the bottom ones, the soybean with the gene and without the gene after spray the herbicide. And you can say without the gene, the whole plants are killed. And also use that method, we can just produce the virus resistance. There are many examples of how we can operate. And we need to find the correct gene and transform the gene into the variety. And their results are very amazing, and you can see from the pictures, with the genes and without the genes in pumpkins, and also without and with, and virus gene in the paper with the genes, and also with the gene and without the gene. So yeah, and also this one to transform a special gene which can give the fruit a longer period of time for storage. And because we know once we have the tomato, and the tomato cannot be stored for a very long time, made normally for just a couple of days. And if you do want to make it longer time for us to sell the fruits, or for example to extend the shelf time, either we can just store the vegetables in a cooler or lower temperature condition, and or we can do the GMO products and to transform some sort of special gene which will give the natural inheritance of elongated storage. And in China in 1998, and people or scientists have already released its GM storage tolerance tomato in Hubei province. And that's the first vegetables with GMO permitted to release in China. And also, in the other part of the world, scientists have already tried to do something with animals. For example, with the fish. And the above one is a normal fish, and the bottom one in China in 2006, people from Zhejiang province, Zhejiang Institute, they introduced some sort of red fluorescent gene into the wild fish and changed the fish body, the color from that one into the red to make it colorful. So that's the very interesting example of GMO fish. And also, we might have already noticed that information or news from the newspaper. At the end of 2015, the USA FDA says okay to the GMO modified salmon fish for human consumption. And months later, Canadian authorities approved that sort of GMO fish, which is considered to be as safe as the wild type fish for human consumption. And in that fish, there are genes which linked to the hormone production and transferred from the other species into that sort of salmon and produce the new variety. And that variety since can produce more hormones and they can grow much faster and bigger. And also some people tried to do something very interesting. They tried to collect some sort of a gene from a cold water fish and to transform that gene into tomato. And in that case, they're supposed to have a new variety with the elongated storage, because at that case, the legal temperature for tomato can be even lower than normal. They can survive even under the water, even with the temperature dropped to 2 degrees. And we all tried some work with the GMO and produced the GMO rapeseed varieties and with the enhanced oil content from the normally 48% to over 50%. And nowadays, believe it or not, the GMO products have already been widely produced throughout the world. And the USA is the #1 country in area for plant GMO crops, and followed by Brazil, and third Argentina, and China is the 6th biggest country for GMO products. That's because most of the people are against GMOs. That makes China is now not on top of the list. And normally, in China, we plant the GMO cotton, just as I said as an example, BT cotton, and also for some trees, and even some papers, and fruits, and trees, and also a fruit which is called papaya, fruits, tropic fruits, okay, and that sort of things. And now China is ranked the 6th, and even this country is on the list as well. So we said the Costa Rica, according to the area of GMO products produced, is listed on the 26th. Because that's because the country is small, so that's no wonder for that. And just as I said, the GMO products in China is now widely opposed by the local people. And normal people, over 90% of the people are against it rather than support it. So that's the reason why we've already received the safety certificates for GMO rice and maize, but they are not yet commercialized in China. And so just summarize, just give you some idea about the advantages and the disadvantages for the different breeding methods. And firstly, the commercial ones, the crossbreeding, the limitations we may know, because that's expensive. We need a lot of labor work and also time consuming. We need to select from the offsprings F1, F2, F3, F4, F4, 5, 6, 8 generations. So that's time consuming. And also that sort of method can transformed gene or induced some sort of variations which are very strictly restricted to the given gene pool because most of the processes are made within a single species. And for mutation breeding, and the limitation is very easy to understand because it's random and highly unpredictable. We are not sure what we can get when we treat a puncture of seeds. We won't say we will have something with higher yield. No, nobody can guarantee that. You can just try to find the differences from the treated seeds. You will not tell what will happen for sure. And for the genetic engineering, it can solve some of the problems because with the gene you transform, you know that's the thing I'm looking for, and the gene once I transform into the plant, that may be there and will be there, and that can be predicted. But the problem is, for genetic engineering breeding, it's highly copy and also variety specific, because that has some sort of techniques involved. We need to culture, and we need to do the in vitro culture, and in vitro culture is highly variety specific. Some of the varieties are easy to regenerate, and for the others, we will find no way to regenerate the plant. And also, the comparison of different breeding techniques. For the commercial breeding, in a piece of land, there's no problem because the breeding work, the end of the breeding work must be in the field. You cannot have a new variety from the test tubes. You must do your trial work in the field and try to find out whether or not your variety or mutant can survive and fit your expectation. And also we need some laborers to do the field work for you. And we do not need quite a lot of money because we don't need very high specific techniques, and we do need money, but we can need a very limited amount of money. But for the technical breeding, we still need a piece of land. Just as I said, you cannot just get a new variety from the test tubes. And we need people to work for you, and also we need technicians or scientists. And we need some instruments because we are doing the technical ones. Without the instruments, we can do nothing. And in that case, we need more money to support the laboratory work. And for the molecular work, still we need the land, we need the people to work for you, and we need well-educated scientists to design or to operate the molecular work for you. So in that case, it is very expensive. So if you have already established your own business in breeding, you need to look into your pocket and try to find out how much money you have. And with that money, you can decide what kind of method you will use. And to make your breeding more profitable and more efficient. So just to give you a very brief timetable of the vegetable breedings in China as a summary for the whole plantation. And also give you a very overall idea about how fast or rapid our development in science and technology. And the new China was founded in 1949. So as you may know, the People's Republic of China established in 1949. And after that, we have around 10 years of very dark period of time. 10 years, that's a very bad history. And that starting from the the late 1970s to early 80s, we restarted everything. We started our science and technology and research. And from that time, the government paid a lot of money and pushed Chinese people or scientists to catch up with the development in many different aspects of science. And then we also did the collection work and screening work started from that stage. And during the 1980s to 90s, we have catch up with the modern or the advanced countries in hybrid breeding and also the biotechnological breeding. And after the 1990s, and the science and technology in China is developing faster and faster. And now we are now leading to the molecular breeding, and in quite a lot of aspects, we are now doing the work which is in the leading position throughout the world. Just give you an example. From 2010 till 2016, the number of very high impact of publications, SCI papers, on vegetable science made by the Chinese scientists. You can see now every year we will have around at least 600 very high impact SCI papers published by Chinese scientists. So that's a very, very huge achievement made by the Chinese scientists. So that's the reason why I just want to give you a very brief introduction of what we are doing and what have already been achieved in China. And from this, you can just try to find out the opportunity for you to establish some sort of certain collaboration between you and the Chinese partners. And now we are in a new era. China has already opened its door to the outside world, and you can easily find the opportunity to establish some sort of collaborative work, relationships with China and also with Chinese government. So here, on behalf of the government, I would like to invite you to visit the country and to get to know more about the country. So that comes to the end of this morning's presentation. Thank you very much for your attention. If I don't make myself clear and you don't have any questions, I don't know. This? No?
Hi, I'm sorry, I arrived a little bit late. I was on a meeting, but from the part that I saw, there is, you mentioned a bit the transgenics. Then in this case, how is the perception in China? Is people consuming transgenics?
Well, that's the question always being asked by people, even in China. As I show you in that picture, in China we already have quite a lot of GMO products growing there. So your answer is yes. Many people have already consumed quite a lot of GMO products. Most the crop with a very high quantity consumed by Chinese people is soybean, because over 90% of soybeans are imported from countries such as America and Canada and the other countries. And over 90% of the soybeans are imported. And we Chinese people must eat that soybean. So we will already consume the GMOs. Okay?
But in that way, when it comes to develop new products in terms of breeding, what is the advice? For instance, if it is for fruit breeding, would you recommend going as using transgenic approaches?
Yeah, in China, that's a very, very, some sort of very rude routine procedure in the breeding programs because you know maybe the breeders won't tell people they are using that sort of method, but indeed, that's the way they can just speed up the breeding procedure and to shorten the breeding time and to make the selection more specific. So that's happening, or that's being done by many, many institutes in China nowadays. So we do not need to mention or tell the other people what we are doing. We're just looking for the results. And is that understandable? Okay.
Talking about mutations, you have shown us different techniques in order to induce the mutation in some kind of different seeds. But can you talk us a little about some cases of that you can they will be successful for the commercial lines? I mean, like that you induce mutation in some species and you get some product that is right now commercialized to the world.
Uh, that's a very good question. But due to differences in our accents, would you please just repeat your question to me again? Okay? Oh, sorry, that's my fault.
Uh, in your presentation, I could learn about different techniques in order to induce mutations in species. So I just wonder if right now uh, there is some product, some seeds, some vegetable seeds that is being produced and commercialized to different in a different location or zone, and it's the result from some induced mutation.
Yeah, I think I've already mentioned in a chart which shows about the mutations, mutant varieties. And well, in your question, you mean whether or not people can buy the mutations. Is that right? Uh, the mutant lines, there's some sort of things called the breeding material or intern material. That's the thing unsuitable because they are not stable. It's just some sort of material or germplasm developed by the breeders and used by the breeders. And that's not the products which can be sold to the other people. If you do have an interest with some sort of a mutant, it is more likely for you to send emails or just come down to that institute and ask the people, and maybe they will give you the mutants you like or you are interested in, free of charge. That's the thing of the academic exchange. I don't think you have the chance to buy any sort of things like mutants. Okay?
Thank you.
Welcome. Mhm.
They are used for search specifically.
Ah, yes, that's right. That's right.
That's not true. Regarding to the molecular breeding, now with the new technology and with the CRISPR, are you using CRISPR in molecular breeding?
Ah, CRISPR is the very, very latest technology in science, in molecular biology. We do have quite a lot of teams or groups are now working on this. And also that sort of information or knowledge have already been used in the breeding. Since I have left the breeding of vegetables for quite a long time, several years, and I'm not quite sure about the next progress, but I'm sure scientists from China are now doing that sort of work. But I cannot give you a very clear idea about their progress and their products. Sorry for that. Okay. Any more things? Well, we're not, it's not limited on science. Anything you're interested in, possibly we can discuss. Maybe about the country or whatever, even the climate. Which as I said, I don't want to push you very hard, and in turn, I don't want to be put by you. So just enjoy the time. It's the last day for your workshop, so you don't want yourself to be sick anyway. All right.
Different to coaching, do you know other method or technique to induce tetraploid in the plant? Different to coaching,
The culture side.
coaching.
Uh-huh.
Do you know another chemical or another technique to induce tetraploid in the plant?
Yeah. That's quite widely used. As I say, we have a lot of varieties bred from that method to produce the seedless watermelons. And that technique is already being used by the host company, the Java company. They have already released several seedless watermelon varieties. So that's a very routine job we can do for the watermelon breeders. There's no problem for that. It's quite easy to operate. Yeah, we have a lot of varieties. Yeah. Okay, no problem. I just need something to help me.
Bueno, muchas gracias por la explicación. Nosotros como universidad en algún momento trabajamos con semillas que fueron irradiadas con rayos gamma, con diferentes tiempos de radiación. Pero este estudio quedó como solo hasta la primera o segunda generación y no vimos mayor cambio respecto a si son más bonitas, más pequeñas, más largas. Entonces, ¿cómo saber hasta qué momento yo tendría que seguir haciendo las evaluaciones en campo como para determinar si puedo tener algún cambio o alguna variedad nueva?
Uh, that's a Spanish question. And yeah, to do the mutagenesis, there are some work we need to do beforehand. For example, first step, we need to try to decide the proper treatment time and also the dosage. You know, different crops will have different tolerance to a certain treatment method, like gamma ray. And from my point of view, I would like to suggest you started with your work following some of the already published papers. Try to find out what what kind of dosage and treatment time they used in their breeding work. And then you tried your work. And even you need to try more treatments and just to make to choose the best treatment procedure for yourself. Because the percentage of mutants appeared following even a certain treatment might be different among varieties. And since you've already started that kind of work, and you grown your seeds, treated seeds into the soil, and you didn't notice any mutants, that's not your fault, and that might be due to the treatment itself is not very successful. And the next step, if you are still keen on doing this sort of work, I would like to suggest you to increase either the treatment time, period of time, or the dosage. I once tried to do quite similar work, to use the I beam of the nitrogen to treat the seeds. And from the treated seeds, I saw quite huge range of mutants with different shape or different, even for the muskmelon, I even noticed 3 cotyledons appeared on top of the one seedling. So that's quite interesting. And so that's my suggestion for you to continue your mutagenesis work. Will that cover all your questions? Okay, thank you.
I don't know if you talk about this gen. I want to know if you have worked in gen modification related with DNA change.
That's a very interesting and very big question. And that's the thing should be answered by the top government officials rather than me. And from my point of view, I have no idea about whether or not that situation will change in the near future. Because fundamentally saying, I'm not quite pretty happy with GMOs. What's the purpose of developing GMOs? To increase the yield? To decrease the use of chemicals? The answers so far are exactly to the opposite. So in that case, I'm not quite convinced of why we should use GMOs. If that's not the only way for us to change our agriculture, to feed our people, well, why just use it? With very strong sort of against from people. I don't think this is a very good idea for my government or even for any government in the world to push forward the use of GMOs. And because their sort of products solves no more questions, but they produced problems or questions to human. Is that right? So that's my opinion. Possibly that's nothing to do with my government. They can do something else, but that's my opinion. Welcome. nice to be here. So, anyway, you do have questions? Do you still we still have time? So no problem for that. If you like, or you can just copy my PPTs, or if you want, you can also have my email address. So if you do want to set up some sort of collaborative work with Chinese or with Chinese scientists, possibly personally, maybe I can help you just to find the right ones. Not me, I just try to find the right people for you, okay? All right. Okay, so dismissed. Okay. Thank you for your attention and for your questions as well. I'm quite sorry, it's now 12 o'clock. I'm sure our lunch must be cooked as ice.
Nada más, una cosa, atención, por favor, que me habían recordado de ayer, de que para mañana lo del acto ya de cierre,